Oh, hey Charlie, I'm going to record you a quick video on what I basically just described to you in my email that I'm writing you. So with DNA, we always have two strands. You have this two strands, and they are full of all of these little complementary base pairs that you've got one over on this side that always pairs with one on that side. And if we kind of zoom in on this part right here, draw that again. You have an A. A always pairs with T on this side. If you have a C, that always pairs with G. If you have a G, that pairs with C. If you have a T, that pairs with A. So that's the way that the DNA pattern, the DNA code goes. However, when we want to make a copy, the way that I talk about this is we have one side that's called our original. Hope you can read my writing. That is our original strand. And then I'm referring to this as our complementary strand. Okay, complementary just means it kind of fits well with it. So let me run through a quick little practice here. I'll use actually different code than what I used on my email to you. So if we had T, C, A, G, C, A. It's 1, 2, 3, 4, 5, 6 bases. If that's our original strand and I asked you for what the complementary strand is, then you'd say, okay, well I got a T, that's going to be A. If I've got a C, that will be G. If I've got an A, that will be T. If I've got a G, that'll be C. If I've got a C, that'll be G. And with an A goes T. So I just determined the complementary strand right there. Now, when you make mRNA, mRNA stands for messenger RNA. That's the message that goes out of the nucleus. Because the DNA is in the nucleus. It goes out of the nucleus. It goes to that thing called the endoplasmic reticulum. It's going to make a protein for us. So this actually gets unzipped. It gets split apart. So I'll break this off. And now we have to make an mRNA copy from this. Well, it should look a lot like that DNA strand. The one difference is that all the T's in RNA are now U's. They use a different base. It's called uracil instead of thymine. So there are no T's, there are only U's. So I'll use my blue here. With A, normally we would have a T, but instead we have U. With G we have C. With T we have A. With C we have G. With G we have C. And with T we have A. So this right here is our mRNA strand. Once again, mRNA is messenger RNA. Now, that will go ahead and leave the nucleus. So we can erase that DNA now. I'm going to grab my red marker. You might have remembered reading about a process called translation. It was in one of those videos I sent to you. This mRNA needs to be read by something called tRNA. That's transfer RNA. Well, what that is, it's transferring amino acids. So UCA gets read by that tRNA. U pairs with A. Let's see if this one's right. U pairs with A. C pairs with G. A pairs with U. G pairs with C. C pairs with G. A pairs with U. Now, here's the confusing part again here. When these three-letter units here come in here and match up with that mRNA, they are carrying an amino acid. You have to look up actually the mRNA code. So UCA, let me grab my codon chart. So here's a codon chart. I've linked one on that email. In case this looks unfamiliar, you also get one of these in the test. You look up U, C, and then A, so UCA, and that gives us serine. So serine just got dropped off by that tRNA, and the next one, CGU came in here and paired up with that GCA. So you look up GCA, that is alanine. So this tRNA brought in and alanine. So the amino acid chain that is building here now starts with serine and then with alanine, ser-alan. So I hope that helps clear up some of that. I will be here tomorrow, which is Friday, or I'll be here on Monday. I'm happy to meet with you and go over that if you need that. Hope the leg's feeling better, bud. Have a great weekend. I'll see you soon. Bye.